Okay, so this is the third and hopefully final video of the cell membrane transports. Last time we left off, we were talking about active transports, and so we go one slide previous. Oops. It talks about active transports essentially use ATP. They're going to require energy. And there are some cells in our body that use up to 40% of their ATP for actively transporting things into or out of the cell. And a lot of that 40% is used for the sodium potassium pump in many cells. So, and that's a sizable 40%, that's a sizable amount of energy just devoted to actively transporting things into and out of the cell. So often when this happens, you are working against concentration gradients. So like it could be electrical, it could be chemical, or it could be the combination, the electrochemical. But if you needed to, basically, if you have a high amount of sodium outside of the cell and a low inside, and then you're pumping sodium back out to that high concentration, so from low to high, that's not the natural tendency. So that will not normally and naturally happen. So you have to put in energy. You have to use ATP to make that work. So we are going to look at probably the best or the most used primary active transport situation, which is the sodium potassium pump. And so the sodium potassium pump is really, really important because we need to maintain a negative internal basically charge inside of the cells and positive outside. So, and that's really important when we think about things like nerve conduction, where you're going to look at depolarization and things like that, which also plays into like muscle contraction and things like that. So anytime you have depolarization of membranes, this is really important for helping maintain those charges. So when we look at this, so here we're going to see, here's our little phospholipid bilayer of our plasma membrane. Here we have this big orange thing is going to be a protein, and it's a protein pump. It's saying carrier protein here because it's going to carry these ions in and out of the cell. So we can see here we're going to have 3 binding sites for sodium and 2 binding sites for potassium. This is not labeled yet because we're not going to look at potassium until after we've changed the shape of our protein. There's also a binding site for ATP. So ATP is going to be required to change the shape of this carrier protein, this protein pump, so that we can move sodium out of the cell and move potassium into the cell. So right now, we can see the series open, and we have these active sites for sodium right here. So the sodium is able to bind to the sodium sites there, and also the ATP adenosine triphosphate there. You can see the adenosine, and then we have our 3 phosphates here. The last 2 are bonded with a high energy bond, and when that breaks, that's going to release energy, and that energy is used to change the shape of this carrier. So 3 little sodium ions are going to bind to the protein carrier here, because right now this active site has high affinity for the sodium, so the sodium is attracted to that, and it wants to bind there. So we can kind of see that. When we looked over here, the ATP is also binding here. When the ATP binds, and our sodium have bound, 3 of them have bound into those 3 sites, the ATP is going to break down. It's going to break into adenosine diphosphate. So it breaks that last bond between the last 2 phosphates, and that releases energy, and this becomes ADP, and then we have our little phosphate there. When that happens, this provides the energy that's going to change the shape of this molecule. When that ATP pump, it changes that shape, it basically kind of makes it go where the sodium was facing the inside. It changes the shape so it's outside, and when it makes that shape change, it changes the site where these sodiums were attached, and it makes it it has less affinity now. So those sodiums naturally want to pop out of there because they're now in an active site that doesn't have the correct fit. So they pop out of there. On the same kind of token, when these have lost their affinity, now these sodium active sites have the correct affinity. So now the potassium sites are here. Sodium's here, potassium is here. These potassium sites have the correct affinity, and potassium wants to attach. There are two active sites for potassium here. So the potassium will attach to these active sites. And when that happens, we're going to see that, why are we going back to the sodium here? Area change shape, sodium transport across, yes, transport across, they release there, it diffuses away. And then our potassiums are going to attach here. And when they attach to that carrier protein, and the phosphate is released here from the little active site that was there, that's going to basically change its shape back, that protein pump shape back into its original shape. And when it does that, once again, the active site here loses its affinity for the potassium, and the potassium is going to jump off because it's not the right shape anymore. So that's going to be bringing in potassium. So when the sodium potassium pump works, it basically pumps 3 sodium ions outside of the cell to an area where sodium is high outside and low inside. So it's pumping against the concentration gradient. After it sends those 3 sodiums out, it pumps 2 potassium in. Remember, circle K, it's high potassium inside the cell, so we're pumping the potassium to the area of high concentration there. So we're pumping those potassium back in. And so you're sending 3 sodium out, 2 potassium in, with the use of 1 molecule of ATP. So it's basically ensuring that we have those concentration gradients maintained, because we need to make sure that we send the sodium back out, so there's high concentration. So then when depolarization happens, sodium can rush in and cause that depolarization. And also, we have to remember that potassium is constantly leaking out of the cell, so we have to send it back in because it's constantly leaking out of the cell. So we're using this protein pump right here to send 3 sodium out and 2 potassium in, and it does require the use of an ATP molecule that's going to provide the energy to change the shape of this protein pump. So, like I said, this is an example of primary active transport. There's also secondary active transport, and a lot of times people are like, okay, they're both active, so they both require ATP. Well, what's the difference between primary and secondary active transport? So when we look at primary active transports, they are directly using ATP to change the shape of that molecule. So a lot of times they talk about ATPase, because they're basically using ATP, breaking that down to change the shape of that pump that changes the shape of those active sites. So the affinity changes, and that's going to allow things to move into and out of the cell. So here they're showing you the sodium potassium pump, 3 sodium out, 2 potassium in. So it's helping to keep those levels of sodium potassium, high and outside of the cell for sodium, high for potassium inside of the cell there. So we can kind of see that. So sodium potassium pump is in most cells. You also see things like the hydrogen potassium ATPase pumps, and those are found in the stomach, where there's an exchange of potassium and hydrogen. There's also calcium ATPase pumps, where it helps to keep a low level of calcium in the cell. So here we can see calcium is being pumped out of the cells there. So we can kind of see that these are all considered primary active transports. Like I said, the ATP molecule is attaching directly to that molecule, that pump, and helping change the shape of that molecule to pump, things into or out of the cell. With that, so the transport protein, so that protein pump, the transport protein acts as both the carrier, because it's taking molecules in or out of the cell, or both in the case of the sodium potassium pump, and it's acting as the enzyme. It acts as the enzyme, the ATPase enzyme, that breaks down the ATP. It converts it from ATP to ADP, which releases that energy. So the ATPase enzyme utilizes or uses ATP to basically alter the binding sites and change the shape of that molecule, and that's going to allow those molecules to enter and exit the cell. So this is from a different picture of the same process. So here, I kind of like this one because the shapes of the active sites actually look a little different, which makes it a little bit easier to understand affinity. So a lot of times when you look at this and you think about, oh, there's potassium inside the cell, there's sodium inside the cell. Well, you're saying the sodium attaches, but the potassium doesn't get involved here. Why doesn't it want to bind here and ride when the sodium goes out? Well, right now you can see the potassium little active sites right here. They are in a low affinity state. Potassium does not fit here, so it's not attracted to those, so it's not going to bind there. Whereas when we look at our sodium active sites there's a high affinity. These are the correct shape for those little sodium ions. They're attracted to them and they're going to attach there. So here we can see that our little ATP, our sodium potassium pump is basically facing, you have the little pump where it's facing towards the inside of the cell. We have the active sites for sodium exposed. The sodium is going to attach. Once that attaches, basically ATP will attach, and when that ATP attaches, it breaks the bond between the second and third phosphate, which releases energy. When it releases that energy, it generates, ATP breaks down, it releases a phosphate from the ADP, and it causes the shape of our sodium potassium pumps to change shape. When it changes shape, the active sites here for our sodium change shape, and now they have a low affinity. So those sodiums pop out. It's kind of like you were in a comfortable chair and then something happened and it is not the right shape anymore. So you get up and get out because it's not comfortable anymore. So our sodium is popping out right here. You notice when this ATP, or when this sodium potassium pump change shape, now our potassium active sites here are in the right configuration. They have a high affinity for potassium at this point. So the potassium on the outside of the cell is attracted to these active sites. It's going to bind there. And when it binds there, the phosphate is going to be removed from our sodium potassium pump. And when that happens, it changes that pump back into its original shape or conformation here. And remember, when it does that, it goes back to its original shape. The original shape, these active sites for the potassium have low affinity for it. So it's like they change back to that low affinity shape. So the potassium hops off. So when you're looking at that sodium potassium pump, it's pumping 3 sodium out and 2 potassium in with the use of 1 ATP that changes the shape of that sodium potassium pump molecule. So we can kind of see that. And like I said, this is really important for establishing that resting membrane potential where you have negative inside the cell, positive outside the cell. And then if something were to happen, if you had a opening of a ligand gated channel or a voltage gated channel or something, then that sodium can rush in and cause depolarization along that cell. So like I said, or it could be a nerve cell, a muscle cell, something like that. But we'll see that process later on as well when we get into those systems. So when we look at the sodium potassium pump primary active transport, because the ATP molecule is binding directly to the pump, the sodium binding sites have a higher affinity or attraction to sodium on basically the intracellular fluid side, so inside of the cell. After the sodium attaches, the ATP that attaches is going to basically cleave the bond between the second and third phosphates, and when it does that, it releases energy. It creates a phosphate that's still attached to the pump, and then the ADP, which is adenosine diphosphate, and it changes the shape of this sodium potassium pump. When it changes that shape, basically it goes from facing the inside of the cell to the outside of the cell. The sodium binding sites, those active sites, change affinity. They have low affinity now, so the sodium pops out, leaves those sites, and basically the sodium is released into the extracellular fluid. Now you'll notice, once again, we're going through this hopefully a couple times and you'll get it, that the potassium binding sites here now are exposed to the extracellular fluid side, and they have a high affinity. They've changed their shape. They have a high affinity for potassium now. So the potassium would be attracted to these binding sites now, and it's facing the outside there. and the potassium is going to attach to those two binding sites there. And when they bind there and the phosphate head that's still attached right here pops off, it's going to change that sodium potassium pump to its original shape. And when it does, it goes back to its original shape. The potassium binding sites are going to lose their affinity. The affinity is going to be lower, so the potassium will hop off. Once again, the sodium binding sites are now in their high affinity position, and so they could accept sodium, and you can begin that process all over again. So we can kind of see how it works to pump 3 sodium molecules outside of the cell and 2 potassium molecules inside of the cell using 1 ATP that directly binds to the sodium potassium pump molecule itself. So that was a primary active transport, because ATP is directly binding to your pump. It's binding to that actual molecule that's changing shape, and that's going to transport against those concentration gradients. Secondary active transport is a little bit different. The ATP doesn't directly bind to the pump. ATP and carriers are both used to maintain a concentration gradient, but the ATP doesn't directly attach to the molecule that's doing the transporting. So when we look at this, here we can see a molecule that's transporting sodium and some other substance. But on here, nowhere do we see ATP involved. What happens is that ATP is used in the sodium potassium pump to pump sodium out. And then when sodium follows its concentration gradient in, the glucose hitches a ride with it, or I mean, this one they're not showing, but whatever the little square is, hitches a ride with it, and it's going to go against its concentration gradient, but it's able to do this because it's going with the sodium, which is following its concentration gradient. But to establish the concentration gradient, you had to use ATP with a different carrier to get that established. So I know it seems like kind of a convoluted kind of thing going on here, but when you think about secondary active transport, you want to think that ATP is used. It's just not directly binding to the molecule that transporter that's moving those particular substances right there. But it is used to establish concentration gradients with a different pump. So it's kind of have to wrap your head around that. So the gradient across the membrane is where the energy was used, rather than phosphorylating the actual molecule that we see here. So when we do this, one substance moves passively in response to the gradient that was produced, and the other substances, like I said, kind of hitching a ride at the same time. All right, so when we look at secondary active transport, that is the same thing again, so we won't say that again. When we look at secondary active transport, we have 2 different types of secondary active transport. We have CoTransport, which is also called symport, and we have countertransport, which is also called antiport. So we're going to look at both of those. So co is together, counter is against. So when we look at CoTransport or symport, that means that you're basically transporting 2 substances in the same direction. One of them will be following its concentration gradient, and one is moving against its concentration gradient, but it's able to go against its concentration gradient without directly using ATP, because of the fact that a concentration gradient, was used or a concentration gradient was made by using ATP to pump something out with a different molecule. So like I said, when we look at this, we know that there's a high amount of sodium outside the cell, low inside the cell. So naturally, sodium wants to rush in and go from high to low concentration. However, how did we establish that high outside concentration? You use the ATP. You use ATP to basically pump the sodium out and the potassium in. So that sodium potassium pump used the ATP directly, made this concentration gradient, and then secondary active transport, this glucose is going against its concentration gradient, but it's moving in because it attaches to the same molecule as the sodium. And when sodium follows its concentration gradient, glucose is kind of hitching a ride. So it goes against the concentration gradient without directly using ATP because of the concentration gradient that was established for the sodium. I know it seems kind of like many steps to get there. So when we look at CoTransport, ion concentration differences across the membrane are used to supply energy for the transport, because you establish those concentration gradients, and that used energy that set up the process that can allow this to go, like to allow the sodium to follow its natural tendency from high to low, and then the glucose goes with it. So the affinity of the carrier is altered not by ATP. So remember when we had that sodium potassium pump, when ATP attached and basically, went from ATP to ADP, that changed the shape of the molecule, changing the affinity of those binding sites. With this, the affinity of those carriers is altered by an ion, but not by ATP directly, because ATP is not attaching to this molecule. So when we look at this with CoTransport, so here's our little CoTransport molecule. If we imagine this open back here, here on the side, we can see it's open. So here, when we look at this, it has an active site for the sodium, an active site for the molecule transporting along with along with the sodium. It could be glucose, it could be some other things. Glucose is a typical one, so I keep on wanting to say glucose, even though they're not identifying it as glucose there. But we can see that there's a strong affinity for the sodium, so it binds, and a strong affinity for this other molecule, so it binds. Then this is going to change shape. It doesn't require ATP, but it will change shape, and it's going to transport the sodium inside, going from high concentration to low concentration inside. So when that changes shape, the sodium, when it changes shape, the affinity for this sodium binding site changes, so it gets a low affinity and it's going to pop off of there. So when that sodium leaves because of the concentration difference, and it pops out of there, that's going to kind of change the shape for that substrate, that other molecule that's being carried in. And so it's going to essentially encourage the other molecule to also exit, because when the sodium exits, leaves that carrier, it changes the shape of that overall molecule. So then that is forced to pop off as well. Because remember, this, like if it was glucose, there's already high glucose inside the cell, so it doesn't naturally want to pop off it, because that would be going to a high concentration. So changing the shape of the overall molecule when the sodium leaves basically forces this CoTransport molecule to also exit the carrier protein and go into the intracellular fluid there. So here, I guess I shouldn't have gone back ahead the picture again. So here we can kind of see our transport protein. This is the one that's actively transported by secondary active transport. The sodium is following its concentration gradient, because remember we have high sodium outside, low inside. So it pops, both of those pop on there. Your molecule is going to change shape. If we go back here and then sodium pops out, it changes the shape of the overall molecule. And then if this were glucose or whatever it was, it would also pop off, because the affinity at this active site would change, forcing it to leave there. So glucose can be carried into the intestinal walls from food sources because sodium is going to travel with it. So like I said, sodium is it would follow the concentration gradients, high sodium outside, low sodium inside. So it's going to go from high to low. Glucose is going to use that CoTransport, so it can travel into the cell along with the sodium. So, but remember, if you're like, okay, so we didn't use ATP to change the shape, but how do we use ATP? ATP was used to actively transport the sodium outside of the cell using that sodium potassium pump. So in a way, not directly, but indirectly, the ATP was used to make the concentration gradient of sodium being higher outside the cell than inside. And then the CoTransport works together. As the sodium follows its concentration from high to low, the glucose is going to kind of hitch a ride with it there, even though it's not following its concentration gradient. So remember, CoTransport, they go together. Symport, CoTransport, they move in or out together. Countertransport or antiport means that you're sending one in and one's going out. So they're going against each other. So you still have two substances being transported, but they're moving in opposite directions. So this one example of that is the sodium proton exchange. So sodium goes in and the hydrogen ion or proton moves out of the cell. So that would be a counter transport or antiport. Anti is like against. So when we look at this, once again, this is a secondary active transport. How does this work? Sodium was actively transported out of the cell using the sodium potassium pump. So that's where the ATP was used, created a concentration gradient so that the sodium wants to move from outside, high concentration outside, to low concentration inside. So it's going to follow the concentration gradient. And when it does that, it's going to send that proton or hydrogen ion out. And so when a sodium goes in, a hydrogen ion goes out. So since one's going out and one's moving in, that would be considered counter transport or antiport because they're moving in opposite directions. But like I said, we don't see any ATP directly binding to that molecule that's pumping, that's using to move those two molecules, or ions in and one out of the cell, because the ATP wasn't directly used with this particular pump or carrier molecule there, but they used it with the sodium potassium pump. So here, sodium is actively transported out of the cell. That would be that primary active transport using the sodium potassium pump. That makes the concentration gradient, that diffusion gradient, where you have high sodium out, low sodium in, so it wants to move in. And then that concentration gradient basically powers the pump right here that's going to move the sodium in and the hydrogen, that proton, out of the cell. Because once again, there's a higher concentration of hydrogen out here than inside, so it requires ATP, but like I said, it's an indirect use of the ATP. The ATP didn't bind specifically to this carrier molecule. All right, so active versus passive transport. So when we look at active versus passive, active requires ATP, passive does not. So cells that pump ions to maintain concentration gradients are going to be using active transports, because you keep on pushing things from a low area of low concentration to an area of high concentration to really establish a high, a even higher concentration gradient between those two areas. If you keep on pumping things out, how do they get back in? Well, remember we talked about how potassium, there are channels or pores that potassium or sodium can leak through. Remember, cells are really leaky to potassium, so potassium is always leaking out, so that sodium potassium pump has to, basically be bringing the potassiums in. The cell is a lot less leaky to sodium, but there are some leak channels with sodium, just not as many. Potassium is really leaky to potassium. Here, this is showing you several different kinds of transport, so we can see primary active transport. So like here, this is a proton pump here. Here we have basically the sodium potassium pump. So we have sodium going out, potassium coming in. Here's a calcium pump here. We have secondary active transports. Here you can see ATP is directly used. They're showing it's directly attaching and used with that transporter or pump molecule there. We look at all these right here, it's secondary active transport. So ATP is used in the process, just not directly with this little carrier pump here. It usually is used over here in primary active transport, and then that establishes those concentration gradients that is used to kind of fuel these secondary active transports. So here we can kind of see some different basically when we look at this, we can see the symport or antiport, or when you're looking at this, you can see the like symport is CoTransport. Symport is what I learned, so I always want to go to symport, but like CoTransport is probably easier. So sodium and amino acids are both moving in, so that would be a CoTransport there. So we have an example of that. The other three we see here are all counter transports or antiports, where you have sodium going in, the proton being taken out, sodium going in, calcium going out. This is bicarbonate and chloride. So bicarbonate going out, chloride going in. So we can kind of see that. Then we can have things like facilitated diffusion. So remember that simple diffusion would go right through the phospholipid bilayer. So if you were small and uncharged or a lipid-based molecule, you can go right through that plasma membrane. If you have a charge or you're a larger molecule or a larger polar molecule, you would have to use some sort of carrier. So facilitated transport, like glucose can go in through, it has to use a carrier to go basically to get into the cell, as do these ion channels. So if you look at sodium, potassium, calcium, and chlorine, they have ion channels that allow them to enter and exit the cell there. And these are all still, since they're using a channel, it's still kind of a facilitated type of movement there, because it is using a helper molecule. So that kind of summarizes, and kind of see some of the differences there. And then as we're looking at things in the digestive system, and we look at things in the nervous system and in other places, the how muscles contract, we will see some of these things again there. All right, so osmosis is the movement of water down its concentration gradient. So probably by the time that you've watched this, hopefully we will completed hopefully both of the labs that kind of use osmosis. We're going to have the dialysis lab and the lab with the red blood cells. I think we'll end up doing the one with the red blood cells first, as long as we don't get any more snow. And then we'll have to do the dialysis tubing lab after that, just because of me having to shift all of these things around. But osmosis is the movement of water, and it's really the diffusion of water. It goes down its concentration gradient from high to low, and it's going to use a special protein pore called an aquaporin. So aqua is referring to water, porin is pore, so it's basically like a water pore. It's, instead of like a sodium pore channel, the aquaporin is for water. So there's various numbers of aquaporins in tissues depending on the need for water in those different tissues. When we look at the movement of water, we're going to talk about osmotic pressure. So basically, osmotic pressure is an indirect measure of the solute concentration in a particular solution. So the greater the solute concentration, the lower the concentration of water. So if you have a lot of solute, that means that you have less water in that particular solution. And so if you have a lot of solute, you're going to have a greater osmotic pressure because you have more things dissolved into the water. So, and that's going to give you a greater tendency of water to flow. So here we can kind of see a little graphic that shows, this is showing pure water. And then this one is a solution that has some sort of solute in it. The little purple dots represent some solute. It could be salt, it could be sugar. Something is dissolved in that water. So here you have a high concentration of water because it's all water. Here you have a lower concentration of water because now some of those water molecules have been replaced with a solute. When we look at osmotic pressure, it's the indirect measure of that solute concentration. And I think I just said all these. The greater the solute concentration, though, basically the lower the water concentration. And then you have a greater osmotic pressure, which is going to be the tendency of water to flow. So with this, if we kind of look at some pictures, so here we have a beaker. It has in it a little semi-permeable membrane. So when we say something is semi-permeable, you know, or sometimes they refer to it as selectively permeable. It means that certain things can cross and other things can't. So we're going to see that if we put solute into one side of this beaker, so we have a little semi-permeable membrane, we put some solute on here. When we look at the two sides, do the sides have equal amounts of solute? If you counted them up, I mean, you can just look at it and you can say, no. You have 3 over here and you have way more than the 3 on this side. So when we look at our little membrane, can you see how small these pores are? Look how big our little solute molecule is. Our solute molecule is not going to be able to cross through the semi-permeable membrane. However, the water molecules, which are much smaller than this, are going to be able to pass through the membrane. So water molecules can move so that you kind of have roughly the same amount of water molecules to solute particles. So if we have this going on here, a situation like this, and water can move, where's the water going to go from? Where do we have a higher concentration of water? So remember, the more solute you have, the less proportional amount of water you have. I know you look at this and you're like, the water levels are equal, but if you looked at water molecules to solute ones here, we have a lower water concentration here and a higher water concentration on this side. So water, much like anything else, is going to go high concentration to low concentration. So this has the high concentration of water compared to this side. So the water will flow from the, basically from the low solute concentration to the high solute concentration, because this is a high water and a low water concentration. So just be careful when you look at, because this is talking about high and low, and this is the concentration of the solute. Because remember, the solute, if the solute could move, you could just make some of these solutes come over here and then it would balance it out. But this semi-permeable membrane does not allow them to pass, but the water can. So the water is going to move towards the high solute concentration. Because I kind of try to think about it as like, they want to have equal ratios of water to solute on each side. So you want them to be equally distributed. So the water is going to go from this side to over here so we can distribute these so they're spaced roughly the same as these. So when we do this, now we can see that the water moved, and I know this is, people are like, oh, can the water do that? This is our little imagination kind of drawing here. It would look a little bit different in real life. So the water moved in here, and so now you can kind of see that these are equally spaced, as are these, and so the ratio of water to solute particles is the same on each side. So it moved from the low solute to the high solute concentration, which was really the high water to low water concentration, until it was equally distributed on both sides of our semi-permeable membrane. We're going to be able to kind of see this when we do our experiment with the dialysis tubing. We're going to have some different sugar solutions in a dialysis tubing bag, and we're going to insert that into beaker of distilled water, and we're going to see what the movement, where the water moves. Does it move from our beaker into the dialysis tubing bag, or does it move the other way? So we're going to be able to kind of see that, and then we're going to see what the effects of having a lower or a higher sugar concentration is. So that's going to be one of our labs where we'll actually kind of be able to visualize what's happening here. But like I said, slightly different because we're going to use the dialysis tubing bags to do that. Now, equal concentrations of solute to solution on each side. So a lot of times we look at how basically solute to water concentrations, how it affects our cells, and we talk about tonicity. So we're going to use 3 different terms for tonicity. We have isotonic, hypotonic, and hypertonic. So if you have an isotonic solution is going to have the same concentration of solutes in it as are inside of your cells. So there would be no net movement of water into or out of the cell. Yes, water will still exit and enter these cells, but there's no directional movement. It isn't all rushing out or all rushing in. So for us, 300 milliosmoles is basically what an isotonic solution would be for human cells. So if you look at this little picture right here, a normal red blood cell is considered to have a biconcave shape. So it's not like it's not like a beach ball. It's not like a circle like this. A normal red blood cell is kind of like a disc shaped, except it's squeezed in a little bit more in the middle. So it's a little bit wider on the edges and a little bit more squeezed in the middle. Let's see if I can draw it. So it's thinner in the middle and thicker on the ends. It's okay. So it's usually thinner here. So here you can kind of see. Can you see how it's almost like you took like that little disc and like squished it in, like almost like you're going to make a donut, but didn't quite push it all the way through. So it's considered biconcave because it dips in on each side. So you can see it kind of dipping in. And some of these you can kind of see. It's kind of hard, but you can kind of see that biconcave shape like that. And that's if you have red blood cells in your body, and then if you put, an isotonic solution would maintain that, would have the same concentration of solute that you would see inside of your cells. So you would have no net movement there. If you put a red blood cell into a hypotonic solution, so hypotonic means that you have very little to no solute in your solution compared to your cell. So in a hypotonic solution, if you have, think about distilled water doesn't have any ions. They've taken out all the ions, all the minerals, anything that's in the water, they've taken that out. So it's just pure water. So if you put a red blood cell that's normally biconcave shaped into pure water, what is going to happen? Well, remember, inside of your cell, you have all sorts of, things in there. There's different ions. There's, you have things, organelles. There's proteins in there. You have stuff dissolved inside of the cytoplasm, that watery kind of part of the inside of your cell. So there's stuff in there. And so if you take that nice biconcave shaped cell and you put it into a hypotonic solution, that cell is going to basically take in water. A whole bunch of water is going to rush into that cell, trying to evenly distribute all those little particles that are inside the cell. So it has basically the same concentration of solute to water as outside of the cell. So what's going to happen if that cell keeps on taking on water? It's going to keep on going. It's going to start to turn into like that beach ball kind of shape, because water is going to be rushing in, trying to make an equal concentration of water to solute inside of the cell and outside of the cell. But if you have pure water, it's just going to keep on rushing in, and that cell's going to blow up into a big old ball, and eventually it's just going to pop. It will rupture because of the fact that they can never reach that equilibrium point, because there is nothing outside of, if you have distilled water. If you had, a slightly, you had a hypotonic solution that had a little bit of dissolved particles in it, but not to the extent of what's inside of the cell, it'll cause that cell to just, like I said, kind of get round and look more like a beach ball instead of it, but it won't necessarily lyse or rupture or explode like that. If you do put it in pure distilled water, it will. A hypertonic solution, if you have a hypertonic solution, that means that solution has tons of solute, has more solute to water than what's inside of your cells. So if you think about a hypertonic solution, you put your little red blood cell into a hypertonic solution that has some, stuff that's dissolved in there. But if you put it into salt water, what's going to happen? Well, there's a ton of salt and all these ions and stuff in the salt water out here, and a lot compared to what's inside your cell. So your cell starts sending water out to kind of distribute that, so that you go from having that biconcave cell into kind of all its water keeps on leaving. So it starts to kind of crinkly, and you get what's called crenation. You see these kind of spiky looking cells right here. These would be kind of showing you what they would look like if you had them in a hypertonic solution. So I don't know how to get rid of that without doing that. so that we can kind of see that when we're looking at the different tonicities of those solutions. So here's a different graphic. This is showing you, here's the normal cell volume. If we put our the cell into basically an isotonic solution, there's the same concentration inside and outside of the cell. You would have, same amount of water going out as is moving in. So the size and shape of your cell, like a tongue twister, will not change. If you put your normal size cell into a hypertonic solution, so now you can see, look at how many particles are in this. So it's tons of particles in here compared to like when you look, before we put this in here, you can kind of see that the concentration is different here. This has many more solutes in it than here. So what happens, our cell is going to basically send water out here so that it will have the same concentration inside and outside of the cell. So now you can see the concentration is the same, but our cell kind of shriveled up or shrunk because it lost water. The opposite happens if you have a hypotonic solution here. When you put your normal cell into a hypotonic solution, basically there are less, like if we look at our normal to here, there are basically less particles here. So it sends water from the outside into the cell, and your cell is gonna kind of blow up. It's gonna get bigger, so the cell swells. I always think if you put a cell into a it has O in it, hypotonic solution, that O means your cell is gonna get big like a circle. It's gonna blow up. So I just remember, that's how, like, the visual that helps me remember when I'm looking at those. So, and when you look at these prefixes, hypo means below, so you have less solute in your solution. Hypertonic solutions mean you have many more solutes than, what's in your normal cell. Iso is the same, so it's equal on each side. So like I said, if you know your prefixes, it makes it a little bit easier. So when you measure osmotic pressure, it's basically the pressure applied to stop the net flow of water. So as water moves, pressure is going to build up basically as that water diffuses down its concentration gradient. And when we look at osmotic pressure, it's the greater the tendency of water to flow, the greater that osmotic pressure. When we look at that osmolarity, so remember molarity was a concentration. So if you had like a 1 molar acid or a 5 molar acid, the 5 molar acid had more moles in it. So it was like more concentrated solution, or more concentrated acid. When you look at osmolarity, that's looking at the total number of solute particles in, basically in that solution. And so there's a difference between, like, if you have a covalent compound like glucose, it's all held together by covalent bonds, versus a ionic compound that is held together by ionic bonding. If you have, so here's glucose uses covalent bonds. When you put glucose into water, glucose does not break down into carbon, hydrogen, and oxygen ions. It stays together as C6H12O6. Like I said, if you have a sugar cube, you have tons of those glucose molecules all kind of like pressed together. So the glucose, the sugar cube will dissolve, but when it gets down to the, like, when you're looking at the molecular compound there, it stays C6H12O6, that like ring shape structure, that stays together. It doesn't break apart in water. So if you have a 1 molar glucose solution, it also has a 1 basically osmol solution, because this doesn't break down. If you have an ionic compound though, when you put like something like table salt into water, like, so table salt is NaCl, that's held together by an ionic bond. When you put that into solution, the sodium and the chlorine get pulled apart, because remember, water is polar, it has those positive and negative regions, so it basically rips the sodium and chlorine apart. So for every 1 glucose or every 1 sodium chloride molecule you put in there, it breaks into 2 particles, it ionizes. So when you look at the osmolarity of that, it's considered a 2 osmolar solution because this would break into 2 things. If you had something like, so if you had like MgCl, and that's a 2 here. So magnesium chloride. So once again, magnesium is a metal, chlorine is a non-metal. So this is held together with an ionic bond. If that went into solution, if you had a 1 molar, like if we had a 1 molar MgCl2 solution, that would be equivalent to 3 osmoles. So, oops, 3 osmolarity because it looks like 30. Sorry. So with that, because this breaks into 1 magnesium and then 2 chlorines instead of just 1 sodium and 1 chlorine. So this would be a 3 osmol solution because it breaks down into those particles. So that's a little bit different when we look at those. So like I said, when you're looking at osmolarity, that's basically the total solute concentration or the number of particles in your solution. And anytime you have an ionic compound, you would count up how many ions it would break into, and that's that, if you had a 1 molar solution, then you would multiply it by that number of ionized particles, and that gives you your osmolarity. If it is a covalent compound like glucose, then it's just the same because those don't ionize when you put them into solution. So when we look at osmotic equilibrium, like I said, with our cells, it's a little bit different. Like our cells will kind of change shape because like if you have an isotonic solution, you have that biconcave shaped red blood cell. If you put it into the hypotonic solution, that cell is gonna basically take on water and it's gonna swell and make a big circular looking shaped cell. If you put that biconcave shape, normal shape red blood cell into a hypertonic solution, it's going to shrivel and get kind of crinkly looking. It's called crenation. If you have something like a plant, sometimes you can kind of see what happens with that because they have a rigid cell wall. And so that's going to prevent their cells from like exploding. So when we look at this, the black line right here, that represents their cell wall. And then this right here, the little green line here, that's like their plasma membrane. So they have a plasma membrane and a cell wall. So it's going to keep their cells from rupturing if they take on too much water. So if you had if you look here, here we can see that there's a space between that. So if you put like basically like plant cells into a hypertonic solution, that plasma membrane is going to pull away from the cell wall, and what happens is your plants will wilt, or the cells will, you see it as wilting on your plants that sit outside of water. But if you took general biology, you can take little like onion cells or Elodea or other some aquatic plants, and you can put their cells onto like a slide. You can put like a salt water solution on them, and what happens is you'll see that plasma membrane pull away from the cell wall. Like I said, that happens if let's say that you have some house plants and you're supposed to water them probably once a week, depending on what it is. If you have a cactus, not that much, but if you have normal house plants, you probably water them once a week. And let's say you forgot. Now it's been a week and a half or 2 weeks. And you look over at your plant and you're like, ooh, not looking so hot. And it's instead of those nice leaves being, kind of firm and fanned out and flat, you look over and they're all like wilted looking. If it loses enough water, it's going to pull away from that cell wall and the plant's going to wilt. When you give it water, that water will go into, through the cell wall into the plasma membrane, and it's going to push that up there. That's called turgor pressure. It basically like pushes, and then, so if you notice that wilted plant and you're like, oh, I don't want to kill it. So you give it some water. And it's not instantaneous, but if you come back in few hours, you'll notice that the plant's starting to perk up, as it were. It will stop looking so wilted, and it's going to start to look more normal. Like I said, if you neglect it long enough, you can water it and it's not going to help it. But you know, if it's just a little past its watering date, you can give it water and it will start to look normal again. And even if you put if you had like a plant cell and you put it into a hypotonic solution, you won't see that plant cell explode like you would a red blood cell, because of the fact that it has that rigid wall that really helps to hold it in place, and it doesn't allow it to rupture, because that wall helps prevent the rupture there, and our cells just don't have that. So here, when we look at osmotic equilibrium, if the pressure of the column of water, the hydrostatic pressure, it's going to equal the tendency of flow of that water. So here we can see the water flowing in and the hydrostatic pressure that's pushing down here. When you reach osmotic equilibrium, it's going to hold steady there. Like I said, if you have a semi-permeable membrane, this could let water coming in here, but you also have that hydrostatic pressure that's pushing down on your column of water there. All right. So another form of, like when we look at moving things into and out of the cell, and this is really an active transport as well. It doesn't necessarily move things against a concentration gradient, but if you change the shape of the plasma membrane, it's going to require energy to change that shape. But we have two ways that are called kind of bulk transport. With that, so bulk transport means you're going to be moving big things. So you may have some things that are just too big to fit in any sort of carrier or channel or anything like that. So the only way to get things into or out of the cell is to use a bulk transport. So these bulk transports are endocytosis and exocytosis. So endo means to bring in, cytosis is cell, exo is to exit, and then cytosis is cell. So endocytosis is going to bring things into the cell. Exocytosis is going to send things out of the cell. So for cellular uptake, bringing in, or removal, sending out of large particles. For example, when you look at white blood cells and, like you look at like something like a mast cell or a neutrophil, they can go through phagocytosis, which just is a specific kind of endocytosis. They can basically engulf a bacteria and bring it in, and then combine that with a lysosome and break down that bacteria. So that would be endocytosis, because they're going to engulf and bring in that bacterial cell. When the cells make proteins, so think about that protein synthesis, and it went through transcription, it went through translation, built the protein, and then the protein got modified and, formed a alpha helix and beta pleated sheet, and then got folded in that tertiary structure, and you have this protein that is destined to leave the cell and go elsewhere. Well, it's really kind of too big to squeeze through a channel or a pore, so how do you get it out of the cell that it got made in? You can use exocytosis you basically form a little vesicle that's going to basically merge with that plasma membrane and secrete its contents to the outside of the cell. That would be exocytosis. So like I said, sometimes endocytosis is bringing things in that the cell's going to use. Sometimes they're bringing in harmful, like in the case of like white blood cells, they can bring in like things like bacteria that they break down to like kill. Exocytosis can release good things like proteins that were made in the cell. It could also release basically cellular detritus, like waste products from breaking down a bacteria. Those could be excreted through exocytosis outside of the cell. So endo and exocytosis there. We also have a couple special kinds of transports on some different kind of membrane connections that are a little bit different and specialized. So when we look at epithelial transports, when we look at the epithelial cells, the joining of those cells is kind of special because they kind of form a barrier. So they have a nice tight junction between them that kind of helps to hold them close to each other. And you see that in in the epithelial tissue. And you're going to see specialized movement of molecules because of those specialized junctions. Because remember, your epithelium helps to keep stuff outside of you outside and the stuff inside in. So you have these nice tight junctions, but that means that things have to move a little bit differently because of the fact that they're the way that they are connected kind of helps to prevent certain things from crossing. And so it makes that transport a little bit different. So when we look at movement of like things like sodium with some sort of other molecule, this CoTransport into the blood, when we look at this, so when you look at this, you would see the lumen side here, that would be the lumen of like an organ. So it could be inside of the stomach, inside of the intestines, things like that. Then you're going to have epithelial tissue. So not only do you have epithelial tissue on the outside, like your skin here, when you think about like your mucous membranes, when you think about the inside of your stomach, the inside of your intestines, that's also epithelial tissue. It's a mucous membrane versus like your keratinized skin that you have on the outside of the surface of your body, but it's still epithelial tissue. So when you have something in, like I said, in your stomach, in your intestines, it has to cross from inside of your intestines through that epithelial cell and then go cross through that epithelial cell to get to the basically the blood side where it can be absorbed. So if you had nutrients, something in the intestines goes through the epithelial cells in your intestines, and then it has to get into the blood because the blood needs to carry those nutrients to other parts of the body. It has to cross, basically from the lumen into the epithelial and then into the blood side there. So here we could see things like sodium, could follow its concentration gradient to get in here. Over here we're seeing that sodium, they're using that an active transport to pump sodium, across. And then over here, this is showing you, since you created these concentration gradients, sodium's going in, substance X follows it. This would be CoTransport here. So this could be like glucose, like sodium with glucose here. Then we can see here that the ATP, the sodium potassium pump was used to pump the sodium back outside of the cell. Oops, didn't mean to do that, back outside of the cell here. And then our substance X, like glucose, could follow its concentration gradient to get into the bloodstream there. So it's a little bit different here because you're going from a lumen side through the epithelial cell and then to that blood. And also sometimes they call it the serosal side as well. So we can kind of see that happening. So we kind of see the movement of those is a little bit different because you're looking at not a lumen side and a blood side and then something passing through that epithelial cell. So like I said, lumen side is the apex of the cell. So sometimes they use apex instead of lumen. Like I said, when we look at the blood side, that will be serosal side. So this is the mucosal surface. Like when you, like I said, if you think about like the body, like sometimes, so you have epithelial, of course, on your skin here that you're seeing everywhere. But then you have to kind of think people are almost kind of like a really long, or I guess, donut. Like you basically have a hole that goes right down the middle of you, because you can, not that you can really do this, but you could take a piece of string and put it in your mouth and then it could come out. I mean, it would be hard to do, but you basically have a tunnel all the way through. You go down your esophagus, through your stomach, through your intestines, and then that string could come out the anus, because you have basically a hole all the way through the middle of you. And so that's lined with epithelial tissue. So that's what that's the mucosal surface here. So this is the side in contact with like food or urine. So when you look at like the bladder, once again, the bladder has epithelial, tissue in it. Then you have the blood side, which is the serosal side. and it has interstitial fluid in it, and this would be over here, this side right here is the basement membrane. So this is the apical side, and this is the basement membrane side when you're looking at cells like this. So like I said, it's a little bit different when you're looking at this, because you have these junctions here and you have these connections which are a little bit different than we have been looking at. When we look at some of these junctions, we also have what's called tight junctions, where it's kind of almost like a zipper, where they kind of interconnect with each other like this. So you can kind of see those tight junctions here. And tight junctions are really important because they have a really tight connection. They allow water to move down its concentration gradient, or its osmotic gradient, but they only are letting that water move through those tight junctions. So like right here, you're seeing that tight junction, so water can move through there. So I kind of think about like, if you think about like, if you have like, or maybe not you, maybe your kids have like a zippered like lunch bag. So you could put in, an apple and a sandwich and some chips and things like that. And then you can put, let's say that you filled all the rest of the space in there with water. That would ruin all your food, but just imagine you poured water in there, so you have water with the other stuff, and then you zip that closed. So the zipper is like your tight junction there, because it's going to, like, if you had that water, I don't know if you kids have ever, their thermos leaked, or you had water in a thermos and the thermos leaked. What happens when you have that zipper? You can have the zipper totally closed, and then all of a sudden, like if they tip their lunchbox certain ways, water, or if they took milk, which is much worse, can be leaking out through that zipper. The apples, the orange, whatever fruit you have in there, your sandwich, your chips, all that's going to stay in your lunchbox, but that water can squeeze through the tight junction, basically like your zipper on that. So it allows, the tight junctions allow water to move, but it prevents other things from moving through those junctions there. So we can kind of see that, and so that's a little tight junction. There's another one right here. Oh, the picture didn't change. So I was like, did I move it? So what happens is you can have osmotic gradients where when sodium is pumped into like interstitial spaces and things like that. So here we can see sodium is coming into the epithelial cell, and then it gets pumped into the blood side over here. The thing about sodium and water is that wherever sodium goes, water tends to follow. So if you're using pumps to move sodium in one direction or another, the water will follow it. So that's going to be a really important premise as we start looking at some of these other, like when we look at the kidney, we'll talk about, okay, water, if sodium is reabsorbed here, what's going to happen to the water? Well, the water is going to follow it, because water tends to follow wherever the sodium goes. So if you are actively pumping sodium into the blood side, then the water is going to follow it. The water is going to follow that, wherever the sodium goes, the water will follow. So we will kind of see that, and that is kind of important when we're looking at these osmotic gradients, because when you actively pump something like sodium somewhere, then water will follow that, so it can change the concentrations based on that. I did that. Okay. All right, so epithelial transport of water. Like I said, how do the epithelial cells transport water? A lot of it is dependent on the diffusion of sodium into the cells, and then once the sodium comes in, you actively are transporting that sodium out of the cell, and so when you maintain that low level or concentration of sodium in the cell, it's going to affect where that water goes, because the water will follow that sodium. So a lot of water transport is sodium dependent. So when we look at this, it's kind of a multiple step process. So sodium diffused into the cell. So like if we go back, wrong way. If we go back, sodium diffuses into the cell. So we had that. I guess I should put this picture on the page here. Then sodium is transported. If we went back here, we use the sodium potassium pump to transport sodium out of the cell. So this is our epithelial cell here. Then you have osmosis of water. So water is going to go from high to low concentration. And also you'll have those like selective barriers, like tight junctions, which are going to allow the water to flow through. And that is going to cause water to move. So you can see this is important in intestinal epithelium. Like I said, we also see some of this in kidneys with where water is moving. They don't look at, I mean, and you do have some tight junctions there as well, but we're seeing it important in places like the GI tract in the urinary system, like when we're looking at the kidneys, we'll see those processes there. So sometimes the movement of one thing is not solely just based on its concentrations. It can be affected by the concentrations of other ions or molecules when we look at those. All right, I think this is our last slide. No, yeah, last slide. So when we look at glandular secretions, they use a kind of a similar process. So when we look at these, they're dependent on chemical signals to increase the secretion of a neural or hormonal signal. So when we look at glands, they're going to get some sort of signal that's going to cause them to secrete something. So you can have endocrine glands or exocrine glands. So when we look at endocrine glands, so those are ductless. So here we can see there's no duct. They're going to secrete from them, and basically it goes into the bloodstream, and then it's going to travel throughout the body till it gets to that target organ. So when it looks at that, but usually when we look at the endocrine glands, so you're thinking about something like pancreas, thyroid, a bunch of these different endocrine glands, they secrete different hormones that there's no duct or tube that connects them to anything. They just secrete outside of themselves, and those secretions go in the blood and they travel through the bloodstream to target organs. If you have an exocrine glands are like your salivary glands. So if you ever think about like eating a lemon, or you're gonna eat a lemon, and you kind of sometimes will feel that pinch right here. You have a parotid salivary gland right here. It produces saliva, and that saliva it basically goes through a tube and it opens into your mouth. So you'll have secretions that'll fall through a tube that'll go into a lumen, or some sort of organ. So, like when we look at stomach cells, we're going to see all sorts of things. You have gastric pits that are secreting all sorts of different kinds of products that go into the lumen of the stomach. Or like if you think of like salivary glands secreting into the saliva into the mouth, or the pancreas is the one that it connects as both an endocrine and an exocrine gland, because your pancreas does have a duct as well, and it can secrete things like pancreatic amylase into your small intestine. So they're going to be secreting different products that will go, either into a lumen in the using a duct like your exocrine glands, or ductless like your endocrine glands that secrete directly into the bloodstream, and then that travels there. So that's a little bit different, but it's using some of that process where it's secreting from the cells, and it has to travel out of the cells and then go into, like I said, into a tube that goes into a lumen, or out of the cell and into kind of like this where we saw going into that blood, into blood vessels there. Oops, going the wrong way. All right. All right, so, oh, hello. Very big. I don't like it when I'm so close to the screen there. So that goes through all of the components of the cell transport. So like I said, you have passive transports, active transports, you have different kinds of things like we talked about, all of the different factors that affect diffusion, things like that. So there's a lot of things in basically in this unit. There is a, like I said, it's still a study guide that goes along with this. There are some extra questions on MyMac, but there's also the quiz, the 10-question quiz on MyMac. With these, a lot of this has been kind of you watching these videos. And those of you who do Zoom all the time, these are probably really no different other than the fact you can pause me and go use the restroom or go get a drink or whatever and not miss anything. For those of you who are normally in class, this is probably a little bit of a different situation for you, but hopefully since you're able to watch them as many times as you want, it won't be too bad for gaining the knowledge for this particular unit. So I'm hoping it's not too bad, but that's basically everything. It covers everything, and we will probably be covering some of these portions in class, but it was really hard for me to gauge how far I might get or what I might have left over. So I just recorded the lecture between these 3 videos. It's the whole, it's everything. So hopefully whatever I can cover in class before I leave, and then when I come back, you know, I can give you some of a review. Hopefully we will get, you know, you will get all that knowledge between all that, and you'll get time to have any questions answered when we have those few classes that I am here for this particular information. So once again, if you have questions, you can always email me and let me know, but make sure you guys do your study guide and go through and do the homework quiz. Remember, you can do that as many times as you want until you get your 10 out of 10, so that you have that information for the exam. But that is all. So hope you guys have a good day.